Amylase is an enzyme that catalyzes the hydrolysis or the breakdown of starches into sugars. It is also known as a digestive enzyme. This is the molecular structure of amylase. This is a diagram of amylase, and in the yellow is the short chain of sugars. Structure. Amylase is composed of amino acids like any other enzyme. It consists of a chain of 5 sugars bound to the active site, and it has a calcium ion which is used to stabilize the structure. The chloride ion is found under the active site to assist in the reaction. The red and white is the active site, which consists of acidic groups. The yellow and orange are the chain of sugars. The gray sphere is the calcium ion, and the green sphere is the chloride ion. History. Amylase was the very first enzyme to be found. It was first discovered and isolated by Enzyme in 1833, where he first called it diastase. He tried producing food from these excess materials. Source. Amylase is found in the pancreas and salivary glands. In the mouth, when you chew, your saliva contains amylase that mixes with the food and activates the breaking down of the starch into monosaccharides. So for example, when you chew Ritz crackers, it makes it mushy so it's easier to swallow and digest. In the pancreas, amylase is produced here, then released into the small intestine where the carbohydrates are broken further down into glucose molecules, then absorbed into the small intestine to use as energy. Where it is used. Amylase can be found in flour. A well-known example is Robin Hood flour. When breaking down the complex starches into simple sugars and flour, it produces properly made bread that has a good flavor and rich colored crust. Amylase also allows the proper rising of the loaf. Amylase is also used in yeast. A well-known example is Fletcherman's yeast. When yeast undergoes fermentation to consume the sugars and starches, it releases carbon dioxide gas. Before it bakes, the yeast allows it to rise, which produces soft and spongy bread.